Assalamu alaikum, dear students. Today we're going to continue, inshallah, the course through the emergency distance learning system, where we are going to record all the lectures for you and upload it in Moodle. Today's lecture is going to be the continuation of transcription. I believe most of the sections have already completed transcriptions, but for those sections who did not complete, I'm going to cover today in this lecture the second half of Unit 2.7, which is transcription. So as you might recall, that the process of creating protein from DNA is made up of two stages: transcription and translation. Transcription is a process of making mRNA from DNA, and translation is a process of making the protein from the mRNA. If you also remember that we have the codon table, these codons are related to the mRNA. So on the mRNA, every 3 nucleotides represent a codon, and every codon represents an amino acid. And we mentioned previously, for example, examples here, leucine is an amino acid, and these two codons code for leucine. So UUA, UUG code for leucine, but also we have leucine over here, and you have these codons CUU, CUC, CUA, CUG. All these are different codons that code for the same amino acid. So we have a rule that for every codon you have to have an amino acid, but not for every amino acid you should have a specific codon. Yani, for example, for leucine, you can have any of these codons. The amino acid leucine can have 1, 2, 3, 4, 5, 6. I believe there's another one somewhere. So all of these codons code for the same amino acid, which is leucine amino acid. However, every codon has to code for the same amino acid. So UUA, and I'll call this in a different color here. So UUA always codes for leucine, but leucine doesn't always code for UUA. Leucine ممكن يكون UUA, UUG, CUU. So leucine has more than one codon. However, the opposite is not correct. It's not the same. So UUA. So as I mentioned, every codon has to code for a specific amino acid. However, not every amino acid codes for a specific codon. So this was just a quick revision of the codon table. This table is very important. You should be able, if I give you a codon, to build the polypeptide chain by translating the codon into amino acids. And we will talk more about it in just a bit. So now we're going to talk about these two stages. So we said transcription is the DNA directed synthesis of mRNA. Transcription is the DNA directed synthesis of mRNA. So it's making mRNA from DNA, and that's what we're going to be covering in today's lecture. So if we have a DNA double strand, like what you see over here, okay, so we're going to use the template, which is always read from the 3' to the 5'. We're going to use the template strand of the DNA to synthesize the mRNA. So we are making mRNA from DNA. تحديدا وين؟ الtemplate strand of the DNA. Remember that the template strand of the DNA is always 3' to 5', whereas the mRNA is always made from the 5' to the 3'. يعني احنا دائما نضيف النوكليوتايد في أي جهة؟ في جهة 3'. So the mRNA grows this way, from 5' to 3'. Then what is transcription? Transcription is making or synthesizing mRNA from DNA. So as you can see over here, there's another schematic representation. So you have the DNA, which is a double-stranded DNA, and on the DNA, of course, we have the genes that are segments of the DNA. And the DNA strand that is used to make mRNA is called the template strand. So we're going to use the template strand of DNA to make mRNA, and the template strand of DNA دائما 3' to 5'. So this blue one here is the DNA. Transcription is making mRNA from DNA. How do we make mRNA from DNA? Always from the 5' to the 3' end. By what? Complementary base pairing. A يقابله U, C يقابله G, C يقابله G مرة ثانية, A يقابله U, A يقابله U, وهكذا. And that's how we synthesize the mRNA from the DNA using what we call complementary base pairing. Now, the synthesis and processing of mRNA, هذا هو تعريف الtranscription. That's the definition of transcription. It is the DNA-directed synthesis of mRNA molecule. So transcription is a DNA-directed synthesis of an mRNA molecule. Now the question here, is the whole template, is the whole DNA template transcribed into mRNA? Is the full DNA transcribed to an mRNA? The answer is no, and let's look into the details. We have something called transcription unit. We have something called transcription unit. What is a transcription unit? It is a stretch or the portion of the DNA that is actually transcribed into mRNA molecule. طيب والبقية إيش تصير فيها? So basically not the whole DNA is converted to mRNA. The part of the DNA that will be converted or transcribed into mRNA is called the transcription unit. Unit معناتها وحدة أو الجزئية. تمام? So this may include more than one gene. So the transcription unit can have more than one gene. So let's look at the photo over here. Where's the transcription unit? It's this one over here. So this is a DNA molecule. This part over here is the transcription unit. Okay? The parts before and after have different roles and different jobs. We're going to talk to you about it in just a minute. Okay, the first part before the transcription unit or upstream of the transcription unit is used to allow the enzyme called RNA polymerase, the enzyme. Okay, it's going to be used for the enzyme to bind to the RNA, to the DNA, to start transcription. And then the terminator at the end, which is this red rectangles over here, that terminator معناتها to stop. Okay, is to stop the process. So before and after the transcription unit, we have parts or segments of the DNA that are used to start the transcription, and we call it promoter, and to end the transcription, and we call it terminator. نيجي نمسح هذيلا مرة ثانية. I think I will repeat this to make it clear. So what is the transcription unit? We said the transcription unit is the part of the DNA that will be converted and transcribed to mRNA. So it's this blue part over here. We call it transcription unit. طيب والأجزاء اللي قبلها وبعدها, these parts over here, this one and this one, what are these? The ones here, this is called the promoter region, which is a region of the DNA that calls and binds to the enzyme called RNA polymerase. And this is the enzyme that is going to convert the DNA into mRNA. And then والجزئية الثانية over here, the terminator. This terminator, which is over here, is a sequence of the DNA that is used to stop the transcription procedure. Now, let's talk about it a little bit more. Let's talk about the promoter and terminator a little bit more. What determines where transcription starts and begins and where it ends? So where do we start transcription and where do we end transcription? These are specific sequences of nucleotides along the DNA. The promoter sequence, okay, اللي حددناها بالأصفر قبل. Okay, what is a promoter sequence? That is a specific sequence of nucleotides on the DNA where the RNA polymerase attaches, the enzyme called RNA polymerase. It looks like an egg. Okay, that RNA polymerase enzyme binds to the DNA to start transcription. Now, if you remember from the previous one, it's not allowing me to go back. So the promoter is a sequence of the DNA that allows the binding of the RNA polymerase. So the enzyme will come, it will bind to the promoter area and start the transcription. And we'll show you that using a video at the end. So the promoter determines which strand of the DNA is used as a template. Like, what about the terminator? What is the terminator? I think this color is too dark. The terminator is the sequence of DNA that signals the end of transcription. So it says stop, خلاص انتهت العملية هنا. Okay, now we have completed transcription. So in very short terms, بكل اختصار, the promoter and the terminator are specific sequences on the DNA that mark the beginning and the end of transcription. Now, this specific enzyme is required for transcription, اللي هو RNA polymerase that looks like an egg. شكله مثل البيضة. Okay, so this enzyme احنا قلنا أي شيء ينتهي بase يعتبر enzyme. Okay, so this is RNA polymerase. Why is it called RNA polymerase? والله لأنه إنزيم اللي يصنع لك RNA. عشان كذا سميناها RNA polymerase. Okay, so RNA polymerase is an enzyme that is used to make RNA from DNA. What does it do? يعني هذا الإنزيم, it is used to make RNA from DNA. How does it do it? The first thing it does, it separates the DNA strands at appropriate points. If you remember, the DNA is a double helix. ملتف حول نفسه صح ال DNA? So what happens is this enzyme comes and it binds to the DNA and it opens it. It separates the two strands from each other. يفتح الشريطين مال ال DNA so that you can start transcription. Then the second step is going to join the RNA nucleotides as they base pair along the DNA template. The best way to see this, to understand this, is to see it. So go on Google, and I highly recommend for transcription and translation lectures that you go on Google and write simple video explaining transcription. Simple video explaining translation. Okay, حال transcription and translation أفضل شيء تشوفوها كشكل فيديو. And we will share some videos on Moodle as well. Please download those videos and watch them. They really help support explaining this lecture. Now remember that when you were adding nucleotides to the RNA, we're always adding it at the 3' end, OH end of the growing mRNA. So the mRNA is growing from 5' to 3' by continuously adding nucleotides to the 3' end. So this is your mRNA here, كذا. هذا 5' and this is 3' مال mRNA صح؟ You're always adding nucleotides to the 3'. ضيف nucleotide 3'. دائما الإضافة والاستطالة والنمو على في جهة ال 3' end. Okay? Oops. Apologies. So الإضافات دائما في ال 3' end of the mRNA. The RNA polymerase will transcribe only a portion of the template. We call it the transcription unit. So the part of the DNA that will be converted to RNA, mRNA نسميها إيش؟ Transcription unit. اليونت اللي يتم تحويلها إلى mRNA. Now, we need to remember that there's always a difference between prokaryotes and eukaryotes. We learned that from day one when we were talking about the hierarchy level of organization. And we said every time we go up the hierarchy, then everything gets more complicated. So bacteria are much simpler than eukaryotes. Transcription and translation is exactly the same. We have some differences between bacteria and eukaryotes because eukaryotes are above the hierarchy level of organization. So we need to know the differences in transcription and translation between bacteria and eukaryotes. إيش أول هذه الفروق؟ First of all, in bacteria, we only have one type of RNA polymerase. محظوظين. عنده نوع واحد فقط وخلاص. بسيط جدا. It's always the case that prokaryotes are much simpler and easier than eukaryotes. So bacteria have one type of RNA polymerase that make all the types of RNA. We know that we have many types of RNA. عندنا mRNA, tRNA, and rRNA. And in bacteria, the same polymerase, the same enzyme, makes all these 3 different RNA molecules. That is not the case with eukaryotes. In eukaryotes, for every type of RNA, you need a different enzyme. So you have 3 types of RNA polymerase. عندنا 3 أنواع من ال RNA polymerase. We call RNA polymerase 1, RNA polymerase 2, and we have RNA polymerase 3. the enzyme that is responsible for making mRNA إيش اسمه من الأنواع الثلاثة هذه؟ اسمه RNA polymerase. أنا مصرة على البنفسجي. اسمه RNA polymerase 2. So RNA polymerase 2 is the name of the enzyme that is used to make mRNA. The other ones, RNase polymerase 1 and 3, are used to make the other kinds of RNA molecules. معي؟ إذا نعيدها على السريع، هي سهلة. Because prokaryotes and eukaryotes can be very different because of the hierarchy level of organization, it is always the case that bacteria have simpler mechanism, less complicated. So in bacteria, you have one type of enzyme called RNA polymerase. That one type of enzyme is responsible for making all the types of RNA we have in the cell. So can mRNA, tRNA, or RNA. Eukaryotes لا معقدين. Okay, the eukaryotes, they have three types of polymerase, three kinds of enzymes. RNA polymerase 1, RNA polymerase 2, or RNA polymerase 3. The name of the RNA polymerase that makes mRNA is called RNA polymerase 2. Okay, I think that is clear. خلينا نعيد الشرح على هذه الرسمة. All right, to make sure that everybody understands, let's have a look at this one. As you can see over here, we have a DNA double strand, and what are we trying to do? We're trying to do the process of transcription, which is making an mRNA from the DNA. How do we do it? This section over here is called transcription unit, which is a part of the DNA that will be converted to mRNA. يعني كم بيكون طول mRNA؟ يبدأ من هنا. There we go. That is going to be the length of our mRNA. From 5' to 3'. Okay? طيب, what happens to the sections over here? This, we said, this is a promoter. Okay? Which is the sequence over here. This is a promoter region. إيش هو البروموتر? It is a sequence of DNA that is used to start transcription. So it is a sequence of DNA that is used to start transcription. نسميها promoter. محفز to promote. Okay. شايفين الانزيم؟ ايش هذا الشكل مثل البيضاوي كذا، شكله مثل البيضة. Okay. هذا هو RNA polymerase 2. This is the enzyme. هذا الانزيم اسمه RNA polymerase. ايش رقمه بالضبط؟ 2 في eukaryotes. So what is this enzyme going to do? It's going to come and it's going to open the strand. يفتح الستراند على أساس إنه يقدر يكون لك mRNA. Because now what we're going to do, we are trying to make the mRNA. We're trying to make the mRNA. All right, by complementary base pairing. So this enzyme over here is going to now open the strand. Okay, it's going to open the strand and start adding nucleotides until it makes your الهدف إنه إحنا نصنع إيش mRNA. So the precise promoter sequence determines which strand of the DNA is the template. So wherever we start, where there's a start point, so we know that this is going to be the template. So look at here, we have something called the start point. Inside the promoter sequence, هذا promoter sequence, inside it there's a little, look at this part over here. شايفين هذا؟ هذا الجزء الصغير موجود داخل البروموتر region. إذا البروموتر region هي المنطقة هذه كامل. نسميها promoter region. It is a sequence of DNA that is used to start transcription. بداخل البروموتر sequence في منطقة تحديدية أكثر. Very specific over here, we call it the start point. It is here. It is at this point where the two DNA strands are going to open. Okay, في هذه النقطة تحديدا يتم فك DNA وفتح DNA. So then now we're going to open the DNA over here and start the transcription process. Okay, and this is what the slide says. Within the promoter is a starting point for the transcription of a gene. Binding site of the RNA polymerase is several dozens nucleotides upstream of the start point. In prokaryotes, the RNA polymerase can recognize and bind directly to the promoter region. إذن لو أجي أمسحها هذه مرة ثانية، هذه نقطة مهمة أريدكم تركزوا معي يا جماعة. Okay, let's say this again. Now, this part here is going to be converted to mRNA. قبلها في جزئية مهمة اسمها promoter. Okay, so what is a promoter? It is a sequence of DNA that is used to bind the enzyme اللي اسمه RNA polymerase 2. شايفين؟ We need the sequence عشان ينادي الانزيم، يقول يا انزيم تعال بايند هنا، تعال ارتبط فيني هنا، تمام؟ على اساس يبدأ عملية الترانسكريبشن. So it's the sequence that's going to call the RNA polymerase to bind here. Why? So that it can start transcription. إنزين الحين إيش شغلة الإنزيم هذا؟ شغلة الإنزيم هذا إنه يفك ال DNA strand، يفتح ال DNA strand, right? So the question is, where is it going to start? Inside the promoter, there's a specific place here. اسمه start point. At this start point here, we're going to open the DNA and start the transcription. طبعا this is very easy بالنسبة للبروكاريوت, yeah? Because in the prokaryotes, ال RNA polymerase ذكي يفهم خلاص إنه هذا promoter يناديه من بعيد يقول تعال ارتبط فيني هنا عشان نبدأ transcription. In the eukaryotes, it's a little bit more complicated. Let's see in the next slide. So that's the starting point and that's the promoter region. Okay. Going back to the question, is the whole template transcribed into mRNA? Is a complete DNA strand converted or transcribed into mRNA? We said no, because the enzyme RNA polymerase only transcribes a specific portion of the DNA template called the transcription unit. So we said the transcription unit is a stretch of DNA that will be converted and transcribed into mRNA, and it can include more than one gene. How does it do it? We have transcription in 3 stages: initiation, which is the start; elongation, which is making the mRNA strand longer and longer; and termination, which is at the end and stopping the process of synthesizing or transcribing the mRNA. We're going to look at these stages one by one. Over here, as you can see, that we have the promoter sequence that we discussed. That's the promoter sequence over here. Within the promoter sequence, we have the start point over here. What's going to happen at the start point? إذا نلاحظ هنا, look at this. What happens at the start point? It opens up. So the enzyme is going to open the double strand of the DNA and going to start adding the nucleotides to make and synthesize the RNA. You can see here, this is called the initiation. This process is called initiation. Okay, so initiation is what? It is the binding of the enzyme to the promoter and then opening the DNA strand at the start point. At the start point, we're going to open the DNA strand and we're going to start adding. هذا هو شوفوا هذا بداية ال mRNA. So that is the beginning of the mRNA strand. So this process is stage number 1, okay, and we call it initiation. البداية, to initiate is to start. So this is number 1, stage number 1. And this can be found figure 17.6 in page 309 for more details. Okay, now that's very easy in prokaryotes. كان واجد سهل صح? Now in eukaryotes, things are always more complicated. In eukaryotes, things are always a little bit more complicated. يعني مثلا, in eukaryotes, the starting point and promoter is not enough to call the enzyme. It needs other things called transcription factors. You can see these 3 balls over here. You can see these 3 balls. These are called هذا 3 أشياء اللي هنا. Okay, هذا ما إنزيمات. لا. These are not enzymes. These are called transcription factors. And we need the transcription factors to bind to the promoter before the enzyme comes and binds to the complex. Okay, so let's see. What do we have in addition here? Here we have something called in the promoter region. نرجع هنا عند البروموتر region. احنا الحين يا جماعة نتكلم عن eukaryotes. We said in eukaryotes things are a little bit more complicated. Okay, because they are up in the hierarchy level of organization. So بالنسبة للبروكاريوت سهل. عندك promoter يجي الانزيم يرتبط فيه ويبدأ يشتغل. في اليوكاريوتس لازم يعقد الموضوع شوية. Okay, كيف؟ In the promoter region over here, we have something additional okay, and I'm going to highlight it in yellow here. It's called the Tata box. Tata box. ليش سموه Tata box؟ عشان كلها TA. Okay, so the Tata box here is found in the promoter region. إيش شغلتها؟ What's the job of this Tata box? It's the job of the Tata box is to call transcription factors. So the Tata box is a specific sequence inside the promoter region, and this Tata box is going to be used to call transcription factors. هي عبارة عن بروتينات. تمام؟ These are proteins, and these proteins are first going to bind. You can see these, they're going to bind to the promoter, وبعدين يجي من okay بيجي sorry, I'm trying to change the color. There we go. Okay, then will come the enzyme RNA polymerase 2 and bind to the promoter. هذا الشكل البيضاوي هو الإنزيم. Okay, إذا إيش الفرق بين الانيشيشن في البروكاريوتس واليوكاريوتس؟ إن البروكاريوتس is really easy. عندك promoter starting point خلاص جاء الإنزيم وارتبط فيه وبدأ شغله. في اليوكاريوتس لا، يبغى له شوية تفاصيل إضافية. إيش هي هذه التفاصيل؟ What do we need for eukaryotes? We need what we call the Tata box. That is going to be used to call the transcription factors, which are proteins that will bind to the promoter, and then will come the RNA polymerase 2 and bind to these transcription factors. So these over here. بحط لكم إياها بالأحمر. These over here are called transcription factors, and then this is the enzyme. So في خطوة تسبق التصاق الإنزيم بالpromoter region بالنسبة للeukaryotes. I hope that's clear. If you needed more details again about this, I highly recommend that you read page 310 in the book for more details. Alternatively, use a video that will explain this in a bit more detail. So what is the difference الآن between the initiation stage? What's the difference in the initiation stage between prokaryotes, eukaryotes? أي واحد أبسط وأسهل؟ طبعا البروkaryotes أسهل. Okay, eukaryotes are always a little bit more complicated. Now, in the prokaryotes, like in bacteria, as an example of prokaryotes, صح؟ قلنا bacteria archaea. So bacteria, what's going to happen? You have an RNA polymerase enzyme, okay, and this enzyme by itself will recognize and bind directly to the promoter. مباشرة يجي الإنزيم ذكي يجي ويلتصق بالpromoter region. In eukaryotes, لا. Okay, now we're going to talk about eukaryotes. Okay, now we're going to talk about eukaryotes. In eukaryotes, what happened? You need something else. You need something called transcription factors. You need transcription factors. Okay, and these attach first to the promoter region. These will attach first to the promoter. That will then help the RNA polymerase 2 to bind to the promoter. Okay, طبعا transcription factors هذيلا كيف يتعرفوا على promoter عن طريق what we call the Tata box, which is a DNA sequence full of TATAAA, and it is found in the promoter region of most eukaryotic genes. إذا عرفنا الآن الفرق في المرحلة الأولى من الترانسكريبشن في كلا البروكاريوتس واليوكاريوتس. البروكاريوت أسهل وأبسط من اليوكاريوت لأنه التصاق مباشر بين الإنزيم والبروموتر ريجن، ولكن اليوكاريوت يحتاج إلى مساعد. هذا المساعد هو التاتا بوكس اللي بينادي لك أول شيء الترانسكريبشن فاكتورز وبعدين بيجي الإنزيم ويلتصق فيه. I hope this will help clarify. That was the first stage, which is called initiation. Now we're going to move to the second stage called elongation. All right, in the elongation stage, what happens? We're going to add nucleotides to the growing chain of the mRNA. This is simple. It's just literally the addition of nucleotides to the growing end, which is the 3' end of the mRNA. Remember that this part here is the newly made RNA. So the red strand, the red one, is the mRNA, and the blue one هنا is the DNA. DNA الأزرق. Okay, the blue one is the DNA. This is the blue one, and this is all DNA. And now we are making mRNA from the DNA. من وين? From the 5' to the 3' end. So what's happening? This enzyme called RNA polymerase 2 that looks like an egg, it opened the DNA strand, and now the second stage, which is called elongation, is just adding nucleotides to the 3' end of the RNA. Again, I recommend you watch a video, okay, and read page 309 in the book. So the RNA polymerase is going to untwist, unfold, open the double helix DNA. تقريبا it opens 10 to 20 bases at a time. So it will open تقريبا 10 or 20. يفتح لك 20 كده 20 حبة. Elongates the mRNA and goes to the next one. The enzyme will add nucleotides to the 3' end of the growing strand. So the growth is always in the 3' end. One. All right. So you can see the DNA template is always read from the 3'. So you are reading from the 3' of the DNA to the 5', and the RNA is always made from 5' to 3'. الإضافات دائما في هذا الجانب 3'N بالنسبة للRNA. Behind the point of RNA synthesis, the double helix will then reform and the RNA molecule will peel away. So when it's finished from this part, the egg-shaped RNA polymerase, the enzyme هذا, it's going to move to the next 10 to 20 bases and it will close it behind it. خلينا نشوف. This is a very nice one that shows initiation and elongation together. So as you can see over here, طبعا ممكن أسألكم مباشرة هذا eukaryote ولا prokaryote? You say this is prokaryote على طول, right? Because you can see that there's no transcription factors. So what happens here? You have the promoter sequence. Inside the promoter sequence, you have the start point. The enzyme is going to come. Okay, the enzyme here is going to come. Okay, and bind to اللي هو الانزيم اللي اسمه RNA polymerase. It's going to bind to the promoter. هذا نسميها initiation. And then what's going to happen here? What did the enzyme do? The enzyme now opened. شايفين كيف فتح ال DNA? It opened the DNA strand and started adding nucleotides to start growing, as you can see here, the RNA. Okay? Elongation. يستمر elongation. You can see here the mRNA. شوف ينمو it's growing the mRNA. It's growing from the DNA. And remember that we are always growing from the 5' to the 3'. إذا النمو والgrowth دائما في أي اتجاه. دائما we're adding nucleotides. We are always adding nucleotides in the growing mRNA in the 3' end. This brings us to the final stage of the transcription, which is called termination. Termination means to end, to finish, to complete. And it's very simple. In the termination, you get to the end of the DNA, where you have a specific sequence of the DNA that says, stop, خلاص, the job is over. And you can see here the enzyme says bye-bye, it leaves, and the termination process, and the transcription process is complete. So transcription proceeds until after the RNA polymerase transcribes the terminator sequence in the DNA. طبعا again, we have a difference between the bacteria and eukaryotes in the termination. If you notice that we have differences in the initiation and termination for bacteria and eukaryotes, you need to know the difference between the two. You need to be able to tell the difference between the إيش الفروق يعني في الترانسكريبشن in eukaryotes and prokaryotes. Now, what are the differences in termination between prokaryotes and eukaryotes? In bacteria, again, it is always simple, easy, and straightforward. The transcription stops right at the terminator sequence, releasing a mature mRNA. يعني when the enzyme reaches the termination sequence, اللي هو sequence of the DNA that says stop. ولما الانزيم يوصل عنده يفهم يقرأ السيكونس يقول اوكي تمام معناتها انتهت العملية. Okay, so it stops there and it releases a mature mRNA. خلاص mRNA الآن جاهز وانتهت عملية الترانسكريبشن. In the eukaryotes دائما احنا قلنا شوية الأمور معقدة. The transcription does not stop at the terminator sequence. يعني يوصل لحد الترمينيتور sequence ويشوف انه يقول له خلاص وقف وهو ما يوقف العملية. What happens? The enzyme RNA polymerase 2 is going to transcribe hundreds of nucleotides past the terminator sequence, releasing something called pre-mRNA. خلينا أراويكم إياها بالصورة. Okay, so here is the difference. عندنا transcription and translation in prokaryotes is coupled. It happens in the same place at the same time. However, in eukaryotes, as you can see here, عندنا هنا النواة. Okay, فنعرف إنه هذه eukaryote. Transcription happens in the nucleus. Translation happens in the cytoplasm. So the two stages of the two processes are separated in time and in space. All right. Now, what happens is that in the RNA processing, in eukaryotes, we have something called RNA processing. Okay. يعني ما يكون لك mRNA مباشرة. DNA يعطيك mRNA و mRNA يعطيك بروتين. لا، في اليوكاريوتس DNA يعطيك شيء اسمه pre-mRNA. mRNA غير ناضج، غير مستوي، غير كامل. Okay. And these pre-mRNA, okay, have sections here, as you can see in yellow, that are later going to be removed. Okay, they're not going to be transcribed into translated into proteins. These additions, هذه الزوائد هذه اللي موجودة على mRNA, they needed to leave the nucleus and go outside in the cytoplasm because the mRNA is going to leave the nucleus, trying to find the ribosome. So it's going to use it to protect itself from hydrolytic enzymes and also binding to the ribosome. I hope you have. I hope this is clear. I'm sorry that we cannot use a video where I'm explaining it with myself. لأنه خايفين على الحجم ماله يكون كبير عليكم. We don't want it to be very heavy. We have been having technical problems uploading the lectures on Moodle because of the size. So I'm going to use my YouTube channel for this phase in order for you to be able to follow the lectures, inshallah. I hope this is clear, and if it is not, I still recommend هذا النوع من المحاضرات. Please يا جماعة هذا النوع من المحاضرات لازم تشوفوا لها فيديو. It's very descriptive. You need to just Google. It'll take you 3 minutes. Okay, so just Google transcription in prokaryotes, transcription in eukaryotes. Watch 2 videos, and trust me, this is going to be much easier when you watch it by video. Thank you very much for listening, and inshallah we'll see you in the next lecture talking about translation and how we can make proteins from the mRNA.